We will continue on to session 3, which will be about bioacoustics and social behavior. We begin with the presentation from Dr. Marina Scheumann. She is from the University of Veterinary Medicine at the Institute of Zoology in Hanover, and she is leading the working group Biocommunication in Mammals. Her main interest is vocal communication and how it can be used as a tool for conservation and animal management. In 2016, she received a grant from the German Research Foundation, which is also called the Deutsche Forschungsgemeinschaft, to study vocal communication in captive white rhinoceroses. So we thank Dr. Marina Scheumann for being here with us today. And if she is ready, she can go ahead and share her screen.
Welcome to my talk. I hope you can see everything. Nobody complained, so I would start. Yes, welcome to my talk. I would like to talk today about our research on vocal and social behavior in white rhinoceros. And as Ido already said, I'm interested in acoustic communication because in the animal voices, a lot of informations are encoded. So you can simply extract by listening to the animal voices in which situation the animal is. So for example, when it's uttering an alarm call, then you know it's in a dangerous situation. When it's uttering a mating call, you know that it's ready to mate. Additionally, you can extract information about the internal situation of the sender, such as physical characteristics of the sender, such as species, sex, or age, information about the emotional and motivational state of the sender, as well as information about the physiological states like health status or reproductive status. And moreover, you can also extract information about social relationships. I think we already listened to or heard in the talk of Julia that we can also extract information about social bonds and social relationships. And also in the animal voices, it can be encoded whether it's a dominant or subdominant animal. And because you can extract so many informations based just by listening and analyzing animal vocalizations, I personally think that bioacoustics, the field of vocal communication, can be an interesting tool for captive management. In our study, we focus on the white rhinoceros. Why the white rhinoceros? At first, it's also a threatened species. We have at the moment 10,000 animals on Earth, and the populations in the wild are declining due to the high poaching, but also to habitat destruction. Therefore, breeding in captivity is still important, especially because studies are done which show that you can also reintroduce captive rhinos back to the wild, and therefore the zoos are a very important genetic reservoir for this species. However, the zoos are not always self-sustaining. We hear today that Hodenhagen has a really good breeding group, but this is rare in Europe, and we have a low reproductive success, and there are several attempts to investigate why is this the case. There are a lot of people which look on the veterinary aspect, especially in Berlin, but we wanted to look on the social behavioral and the vocal aspects. And the white rhinoceros is a little bit special from other rhinoceros species, and therefore also suited for this kind of analysis. Because in contrast to the other rhino species, we have here a so-called semi-social system. That means the males live solitary and have their own territories, but the home range of the females can overlap with several males. And what is so special is that the females and the subadults can form temporarily stable groups with up to 10 individuals. When we started the study, there were already 2 publications on the vocal repertoire of the species available. One doctoral dissertation by Owen Smith, and I have to say this is the Bible for all rhino people. And another publication by Roland Pollicht, who studied the last herd of the northern white rhinoceros. I have to say that this subspecies, there's only 2 animals available on Earth. These are 2 females, so we can say that this subspecies is almost extinct. Both studies showed 10 to 11 different call types, but when you have a closer look, you will see that they correspond only in 5 call types, whereas the other call types were either observed by Owen Smith or observed by Pollicht. And that means that even if we have 2 studies, we need more information to clarify the vocal repertoire of the species, which is, of course, the basic for further research. The the call type which was best studied in rhinos is the so-called pond call. There are very nice study by Ivana Zinkova, who already found out that the pond call can encode gender identity, age, sex, dominance, and motivation. And here we add our studies, and today I would like to talk about 4 topics we are investigating. At first I want to show you a short overview about the adult vocal repertoire and have a closer look to the infant vocal repertoire. In the second part, we investigated whether next to the pond also other call types can encode for gender identity. And then we was interested whether the vocalization can reflect social relationships between sexes. And last but not least, we was interested whether vocal and social behavior can be used as indicator for female receptivity. Where do we work? Today I will talk about our captive studies. So we were in 11 zoological institutions. We visited all zoos in Germany, and afterwards we expanded our research to France and to Denmark. And in total we observed 53 different subjects. And here I would like to acknowledge my 2 PhD students, which did the main work, and the data I present today are parts of their PhD thesis, which they hopefully will finish in the next month. And this is Julia Janikeev and Sabrina Lin. And what you can see on the pictures very clearly, this is our main equipment. This is a microphone, and we have, of course, a camcorder. And the real work starts at first when we are back to the lab. Then we do the behavioral observations by video analysis and analyze also the vocal recording using specialized software, which helps us to make a mathematically description of the calls. To have a short look on the vocal repertoire, the repertoire of the rhinos contains mainly noisy calls. One call type, the other in aggressive situations, when as a kind of first warning, is the so-called hiss call. Another call type when they are really fighting, so when they have really strong aggression and have horn clashing or really physical interactions. This is the grunt. A call type, the other during feeding and resting, is the snort. And the call type other during mating or when they try to approach each other and search for contact is the pant. And we also found the whine call, which is mainly uttered by infants and subadults. With this, we could confirm the 5 call types where the previous study already correspond. We additionally found a call type which was reported for wild rhinos, but not for captive animals, and this is the so-called squeal. And when you now think this is an infant because it looks very similar and it's very high-pitched, then you're wrong. This is a call a really big adult bull is producing, and it's suggested that they produce it when they try to prevent that the females leave the territory. And this call shows very clearly how important it is not to observe only one group or 2 or 3 zoos, because this call type we observed only in two zoos which we visited. So it's a call which is not so common in zoo populations. What you might notice is that there are less calls than 10 to 11. This can have different reasons. At first, maybe in captivity, we observe not all call types than we can observe in the wild. But the major impact has here the definition of a call type. We defined as a call type when he is acoustically different, but also when he occurs in different behavioral contexts. And this is different to the studies which were done before. To explain you what I mean, I want to have a closer look with you to the infant vocal repertoire. So for example, these are typical infant calls the infants produce, and they vary a little bit. You can have short and high-pitched calls. You can have short and low-pitched calls. You can have long and high-pitched calls. And you can have long and low-pitched calls. And in the previous publication, they would be classified as two different subtypes. Their own Smith and Polished would classify the wine A and the wine C as a squeak call, and the wine B and the wine D as a whine call. Why do we not do this? The reason is very simple. All the calls occur in a similar context, so they all occur during begging, during nursing, and when the female wants to have, when the infant wants to have the attention of the mother, and therefore we see no differences in the behavioral context, and therefore we classify this as one call type. And here maybe further study is needed to investigate whether maybe the different variations which can mix up and which show also strong transitions to each other might reflect other information such as arousal or, for example, the level of hunger. When we further looked to the infant vocal repertoire, we could also observe 3 adult calls. This is the snort, the hiss, and the pant. We did not observe grunt and squeal, but this is not so surprising because infants have almost nothing strong agonistic reactions or have not to defend their territory. Therefore, it's not so surprising that these call types were not uttered by the infants. We had thereby the possibility to also investigate an hand-reared calf, and we observed that this hand-reared calf, which has no physical contact to other rhinos, produced the same number of call types and produced these call types also in a similar context. And therefore, we suggest that the vocal repertoire of the rhinos is innate. We then was interested to look on whether sender identity can be encoded in the different calls. I already mentioned that for the pant, it was also already known by Zinkhofer and Polished, and we measured it additionally for 3 other call types, especially because the snort and the hiss are the call types which you hear much more often than the pant. We calculated the so-called information criterion. This is a criteria which is relatively robust against different sample size and different number of parameters. It's very complicated to calculate this criterion, but what is for you important is that as higher the value is, as more individuals you would be able to discriminate. And what you can see is that indeed the reported value for the pant was higher. We found a little bit a lower value for the grunt. And when we look to the hiss and the snort, then the values increased dramatically. And what you can also see here that the calls were produced in different contexts. And with our data, we could support the social function hypothesis for individual distinctiveness. Thus, calls uttered in affiliative context show a higher individual distinctiveness than calls uttered in agonistic context, whereas calls uttered in agonistic context have a higher degree of individual distinctiveness than calls uttered in non-social context. And with non-social, I mean feeding or alarm calling. Then we were interested to look whether these calls might be able to give us some insight into the social relationships, especially between the sexes. And for that, we used vocal network analysis. And we calculated sender and receiver diets for that, the call rate for sender and receiver diets for that. Now you have one problem in rhinos. A rhino is calling and nobody reacts. So it's not quite easy to find the receiver based on behavioral differences. And the other problem is even if the rhino is not react, it can still be a potential receiver. Therefore, we used a proximity measurement to determine the potential receiver, and we used the proximity of 1 body length. That means all animals which were in the range of 1 body length to the sender were determined as potential receiver. And for this sender-receiver diets, we calculated a dyadic call rate, meaning the number of calls per hour contact time. Based on that, we constructed vocal networks, and the vocal networks look like here. You have their knots that could be either the squares are males, the circles are females, the knots represent the subjects. Then you have here connections between the knots, which are called ties, and the thickness of the ties signals the call rate, and the color of the ties signals who is the sender. That means in this example, the pink female emits a high number of calls to this male, where it emits only a low number of calls to the blue female. By plotting this, we found a really distinct structure. So you see it's not randomly distributed. You have really diets where you have a high vocal exchange and diets where you have a low vocal exchange. And we tested it also for symmetry, but this exchange is asymmetrical. That means when you send a lot of calls out, it does not mean that you also get a lot of calls back. What you can also see here in an example for the his and the pans, that the vocal networks look quite different for the different call types. And what I also want to show very shortly is that this is not a case this is nothing what we've observed in one zoos. This is a pattern, and this is very important for us that we do not report case studies. This is a pattern we really observed in all zoos we are visiting. To look for sex-specific vocal asymmetry, we use the in-degree and out-degree weights. And the in-degree weight mean that it's the sum of the call rates the animal received. So that would be a green plus pink plus blue. And the out-degree weights would be the sum of call rates the animal emitted. In this case of the male, he emits nothing of this call rate. Based on that, we calculate an index which ranged from -1 to 1. -1 indicated that the subject produced more than it received. 1 indicated that it received more than it produced. Based on that, we compared it for males and females and found indeed sex-specific differences. Thereby, males receive more his than they emit in comparison to female, whereas they emit more pans than they received in comparison to a female. We were further interested to look whether this is only an effect of the sender or whether also the recipient has any influence on that. And by that, we calculated linear mixed effect models testing for diet type and for interaction type. And indeed, we found an effect for the his. That means that females emit much more his to males than they would emit to other females, or than males would emit to females. And we also found that the his rate correlates with the aggressive interaction rate, whereas we found no correlation with defensive and affiliative interaction rate. For the pans, we could only found tendencies which might be related to the lower sample size. Pans, when we recorded pans not so often in captivity, but also here we found a tendency that the diet type has an effect. In that case, it's the male who utter more pans, what you can also here see very well, which utter more pans to the females than females utter to males or females utter to females. And here we also found a tendency for a correlation, in this case with the defensive interaction rate, which shows that we have a sex-specific vocal usage in the species. We then was interested whether we can use it maybe to have indicator of the receptive phase of the animal. So we collected not only audio and video data, we also collected fecal data, and this fecal data were analyzed in collaboration with Jella Wouters and Martin Dehnhardt at the Institute of Zoo and Wildlife Research in Berlin. We analyzed all 3 sex steroids or metabolites of that, progesterone metabolites, estrogen and testosterone metabolites, but I will only present data on the progesterone metabolites. We had a sufficient sample for 15 animals, and what we found corresponds with the literature that at least 50% of our animals do not cycle, and this corresponds to the low reproduction rate. And in 7 animals, we could found cycling activity, and in 1 animal, it turned later on out that she was pregnant. In the majority of cases, we really found mating behavior corresponds to the hormonal cycle, but we had also 2 exceptional cases. In 1 case where the female showed no mating behavior, but we saw at the end a hormonal cycle, so an increase of progesterone values. And in 1 case where we observed mating behavior, but we found no hormonal cycle when we looked on the progesterone values. Therefore, and also because we found no reliable measurement to use the hormone data to calculate the receptive phase, we come back to the old good parameters mating and copulation to define the receptive phase on an animal, and we calculate the day before and the day afterwards to that. And we compared it with the day 3 and 6 before and after the receptive phase. And this we could do for 8 male-female diets. And what was not surprising was that we found differences in the social behavior between the different time intervals. So in both sexes, the time they spend with each other, social cohesion, as well as the rate of affiliative interaction increases at day 0. Interestingly, when we looked to the vocal and olfactory behavior, we found differences, whereas the males increased their pant and hiss rate at day 0, we found not such an effect for the females. Instead, the females increased their olfactory behavior, showing more marking behavior. They have also a special marking behavior, that's urine spraying, where they really spray their urine out, and showed also more presenting behavior. And the marking behavior, for example, or the olfactory behavior in males was not changed. So that we found that there seems to be a sex-specific signaling of mating motivation. To sum up, what we can say today is we can confirm 6 call types for the captive white rhinoceros. We suggested that the vocal repertoire is innate. We found to a lower degree individual distinctiveness in 3 more call types, but really to a lower degree except for the grunts. And we could support that social calls exhibit a higher individual distinctiveness than calls emitted in non-social contexts. We also found a distinct communication network with sex-specific vocal usage, that males utter pants towards females, which correlate with their defensive behavior, whereas females utter hisses towards males, mainly correlating with aggressive behavior. And we also found a sex-specific signaling of the motivation to mate, whereas males seems to use more vocal signals, females seems to use more olfactory signals. What we plan for the future, or what we have already studied, was playback studies to confirm that the call types will also perceive differently. We already did a pilot study in France, and then Corona came, and we had to stop last year because I think we all know that the zoos have at the moment different problems than research. We are also interested to go further and to look whether the sex-specific signaling can maybe be used to facilitate mating behavior. And we already started a study in the wild. Here you can see a further PhD student, Daniela Lan, which recorded rhinos in Uganda. And we are also collaborating with people working in Botswana and South Africa to get a much more clear picture on the vocal repertoire, but also to get an idea whether the animals in captivity show their normal behavior. And last but not least, research. We all know research is always teamwork, and I would like to thank my teams and my collaborators, not only the PhD students, but also all the master and bachelor students which contribute to the project, and the funding agency, and of course Jella and Martin for their hormone expertises. And what I especially want to emphasize is my thank to all the zoos which participated and support us long before we got the money from the funding agencies, just by free accommodation. And I just want to emphasize that this is very important to push up zoo research. And with that, I and my rhinos, or the rhinos, would like to thank you for your interest and your attention. And it's really a pity that we cannot see them. I know a game drive was planned in Hodenhagen, but at least you can look on the enclosure of them.
Can you see the questions on there?
This is okay. The first question is his dissertation, and if you're interested to read it, there is a really cool page. The Rhino Research Center, a guy in Vienna collects everything about rhinos, and there you can also have a PDF of this Bible. And the second question: it is known how far their calls travel. Yeah, it's a little bit tricky to know. I know that the question maybe relies to our definition of the proximity. This was mainly based to logistical reasons, I have to say. A lot of the calls are very soft, and the pan call is suggested to be a long distance call, but I'm a little bit in doubt of it because it was really hard for us to record them in sufficient quality. But directly known how long they go, I think there's no study at the moment.
Okay. Would anyone have any other questions? You can also raise your hand, or you can unmute your audio to ask her directly. If you do not, then I have an announcement. I'll just wait a second in case anyone has any additional questions. I will stop my screen share.
In fact, there is another question in the chat area.
Okay. So this question is it possible that white rhinoceros can also have a distinct call if they are suffering from a disease which is not usually displayed by the animal? I was just thinking that it could possibly help in disease diagnosis or something.
That could be. So what we know from other animals and even from ourselves, when we have a cold, then our voice will change. And when if it can affect also the voice of the animals, and I know from a lot of farm animal studies, for example, in pigs, the coughing is monitored to detect whether diseases occur in pig farming, for example. And this is also something we would like to go forward, how animal voices can be used as a diagnostic tool. Especially, you all know Alexa, Zira, and Cortana. And of course, this is also only a mathematically analysis of vocalizations. And I think this can also be transferred to animal voices. And then it might be possible to use it also as a diagnostic tool.
Okay. Does anybody have any other questions? Again, you can unmute your audio or type it in. Okay, so our next speaker is Dr. Angela Suja. Unfortunately, we had received a message that she had a health emergency, so she will not be able to join us for this talk. So what we would want to do is thank you very much, Dr. Marina Scheumann, for your incredible presentation, and we will then take a 35-minute refreshment break. So if you need to stretch out, I know you probably just did that, but if you need to stretch out or grab a cup of coffee or some tea or make some phone calls, you may do that at this moment. Thank you so much. All right. Welcome back, everybody. We will now continue on with the presentations within our session 3. The next set of presentations will be by Ricardo Lemos de Figueiredo. I really hope I'm saying that correctly. He is a PhD student at the University of Birmingham, and his focus is on zoo animal behavior, welfare, and conservation. So he's going to have a symposium presentation for 15 minutes and a poster presentation of 10 minutes. So while that's happening, as usual, please feel free to type in your questions at the chat, to which we can then ask him after his presentations. All right, Ricardo, whenever you're ready.
Okay, thank you. Let me just try. This is my first time using Teams, so let's hope I can share. Okay. Is that right?
Perfect.
Yeah,
yes.
Okay. So hello, everyone. As said, I'm Ricardo. I'm a PhD student at the University of Birmingham, and today I will be presenting about my research work on how to encourage natural behaviors in captive parrots, and starting with the Enclosure Design Tool, or EDT, which is a web application created by the University of Birmingham and partners. And the idea is to help captive facilities like zoos and sanctuaries comparing the behavior of their animals with wild conspecifics and encouraging natural behaviors in their animals. And the way you do it is you collect behavioral data on the captive animals, you upload those data into the EDT, and then the EDT returns a comparison between captive animals and the available data from the wild, and tells you what natural behaviors are missing or underrepresented, and then suggests enclosure modifications to encourage those specific behaviors. The EDT is already up and running for great apes, and the next step is an extension to parrots. Parrots, by parrots I mean all the birds of the order Psittaciformes. They are very popular in human care, with over 60,000 individuals reported in ZIMS in 2018. Over a quarter of all parrot species are threatened with extinction, with the main threats being habitat loss and the illegal pet trade. They are very cognitively advanced. In the wild, they live in very complex environments where they travel long distances between different sites for different activities. And in captivity, they often develop undesirable behaviors like stereotypies, feather picking, which may indicate poor welfare. Captive breeding may be part of the solution to help all those threatened species are often managed in insurance populations in zoos all over the world. They are part of reintroduction programs. So captive breeding can really play a big role in the conservation efforts. However, the problems are that long-term captivity can cause loss and modification of natural behaviors. It can cause changes in organ, skeleton, muscles of the captive individuals. And the success of reintroduction programs really depend on the behavior of the captive animals. So why do we want to encourage natural behaviors in captive parrots? On one hand, we want to maximize the conservation potential of captive parrots by maintaining a natural repertoire of behaviors in the captive population, by keeping the captive population as morphologically similar to their wild counterparts as possible, and facilitating any potential future reintroduction programs that may occur in the future. From a welfare perspective, captive animals must be given the opportunity to express natural behaviors. So by stimulating them and encouraging natural behaviors, we're also positively influencing their welfare. Note that animal welfare science is a very complex topic, requires many indicators and a multidisciplinary approach, of course, but they just fall outside the scope of this study. We focus on natural behaviors with this research. Methods and partners. So we are partnered with Jurong Bird Park, which is part of Wildlife Reserve Singapore, and with Drayton Manor Theme Park and Zoo in the UK. The species that we included in this study are the scarlet macaw from Central and South America, the African grey parrot from Africa, of course, and the swift parrot from Australia. Protocol. So we use different protocols that kind of merge together. In all these protocols, we collect data on general behaviors and enclosure use. Then for the scarlet macaws and the African grey parrots, we have a cognition, a locomotion, and a social protocol. In the cognition protocol, we collect data on the type of manipulation, so how they interact with objects and food, and the type of object and food they interact with. In the locomotion protocol, type of locomotion, flight rates, and the type of supports they use within the enclosure. In the social protocol, how they interact with other individuals and how close or far they stick to the other individuals in the enclosure. For the swift parrot, we have a smaller case study for them because we have a small sample size, but we're doing a foraging study, so how they feed and forage around the enclosure. So just an idea of the results we've got so far, and this is a general activity budget for the scarlet macaws at both zoos and for the groups of grey parrots. Here on the left, I'm just going to put the laser pointer. Here on the left, we've got the scarlet macaws. To the right of the line, we've got the available data from the wild. Note that this is captive bred, released individuals and juvenile scarlet macaws in the wild, which is not ideal for comparison, but so far, what we could find. So this is for cautious comparison. In the graph B, we've got both groups of grey parrots at both zoos, and we've got data from the yellow front parrot in the wild, which is closely related to the African grey, and they share a lot of ecology features with the species. And what we can get out of these graphs, just a general idea, is that all captive individuals spend a lot of time just perching or inactive, and more time inactive than the individuals in the wild or the available data from the wild. The captive individuals generally spend less time engaging in social behaviors and feeding than the wild counterparts, even when we combine feeding and object manipulation, which in captivity can have similar purposes sometimes. Even combined, it's far less frequent than in the wild counterparts, and the same can be observed in the grey parrots. In terms of enclosure use, this is just some examples from some of the individuals. And what we can see is that enclosure use was very uneven. So some areas were heavily used and preferred by the parrots, while other sections were heavily ignored. Some individuals spent too much time on the ground. Some individuals ignored the top section of the enclosure. So there are some opportunities to improve in terms of enclosure use. Food and object manipulation, and the main thing we noticed was that advanced manipulation, and by that we mean really complex, challenging manipulation like puzzle feeders or opening a really tough nut, for example. And that advanced manipulation was very infrequent, quite rare. Was in some individuals, it was actually 0%. And for food manipulation in some scarlet macaws, there was a little bit of it, but it was not frequent at all. Most of the objects the parrots interacted with were permanent and destructible, meaning that they're providing low novelty and low challenge to the animals. In terms of food use, again, we noticed that it was quite rare across individuals. For the scarlet macaws, there was a little bit of it in some individuals of both food and object manipulation. For the gray parrots, food manipulation, we rarely observed food use in food manipulation, a bit more in object manipulation, but it was not predominant at all. And note that food use is a very important natural behavior for these animals in the wild. In terms of locomotion, looking first at flight rates here on the left, again, number of flights per hour was not very high. It didn't exceed 12 flights in any of the individuals. A couple of the individuals did not fly at all, which is, of course, not very normal. And most of the flights observed were under 6 meters, so very short flights. In terms of support use, we noticed a lot of wire mesh use in some individuals, a lot of ground use in some of the individuals, and a lot of stable perch use in a lot of the individuals. Most of the locomotion observed was tripod locomotion, so climbing and descending, but most of these was on the wire mesh. We also observed some walking, and most of the perches used were horizontal or thick. So there was not a lot of diversity in support use. Social behavior and proximity, we observed that from the activity budgets before, that social behaviors were quite low in general. Within social behaviors, we noticed a lot of hollow preening in the scarlet macaws. The African greys, there was more nibbling when they kind of go for nibbles on each other. And we did not see hollow preening in one of the groups of gray parrots, which was quite weird. In terms of proximity, most of the individuals did not spend a lot of time in close proximity. And by close proximity, we mean within one wing length of others, with the exception of the pair of adult scarlet macaws at one of the zoos, because they were a bonded pair. They even bred last year. So they spend, of course, a lot of time really close to each other. All the others, not so much, which means that bonding may be quite low in those groups. With that said, all individuals spend most of the time within 2 meters of at least part of the group. So it's not like they're completely isolated from the group, they just avoid close proximity. So discussion, what can be improved based on these results? One of the things we can improve for sure is increase activity levels. As we mentioned, inactivity was quite high across individuals, and this may mean it may be a potential sign of a negative state. It may be that they're not having motivation to engage in other behaviors. It may be an alternative response to stereotypy, so something to improve. We want to maximize enclosure use, because if we have an enclosure, we want them to use as much of it as possible. We want to decrease ground use in some of the individuals because both species are arboreal in the wild. Wild gray parrots have seen foraging on the ground in the wild, but it is not predominant at all. They're both arboreal. We want to increase the use of the top of the enclosure in both species because in the wild, both African greys and scarlet macaws prefer the safety of the height. So really stay really high on the tree canopy. So we want to do that as much as possible in captivity. We want to increase manipulation challenges. In the wild, they really have to use their cognitive abilities, their big strength to access food, either tough nuts, to access the nuts, to crack open the nuts and seeds. So we want to replicate that in captivity as much as possible. We want to increase the novelty and toughness of objects provided for manipulation, because again, in the wild, that's something they experience quite often. We definitely want to increase food use. Like I said, this is a very important behavior for parrots in the wild. They actually have a specially adapted feet, just not only to climb, but also to manipulate food items. So we want them to use their feet as much as possible when manipulating food and objects in captivity. We want to increase flight rate. In the wild, both scarlet macaws and gray parrots travel. In the morning alone, they're estimated to travel 10, 15, 20 kilometers just in the morning. In captivity, of course, this is very hard to replicate, but what we can do is try to maximize flights as much as possible. We want to decrease wire mesh use because wire mesh is a very boring structure, let's put it that way. It's very stable, it's very homogeneous, doesn't provide the challenges that structures in the wild do. So we want them to use less wire mesh. And we want them to use more diversity of perches. So we want a greater diversity of perch use in terms of thickness, orientation, stability, to kind of replicate a little bit the complexities of the wild environment. And this may be a bit more harder to do based on the modifications we can propose, but strengthen social bonding. We didn't see in some animals, we didn't see a lot of social interactions. We didn't see a lot of close proximity, which may mean that, they're not really bonded with other individuals. There could be several reasons for that. That may be hard to target with the modifications, but it would be something nice to see improved. This is just an example of some of the modifications proposed. Note that these have not been evaluated yet because we have not collected the post-modification data. It's just an overall idea of what we've proposed. And we propose things like high complexity of perches in the corners of the enclosure while providing clear areas in the middle for flight. We propose putting some rope or branches on certain areas of the mesh so they have to climb on more challenging stretches instead of the mesh. More complex feeders. And this is just an idea. Once we have all the data, we will be able to provide more specific recommendations in terms of modifications. And just an overall idea of at what stage I'm on with this project. We've collected baseline data, but we're finishing for the swift parrots, and that's why they've not been included in this presentation. We have to collect post-modification data for all the species, and then we will, of course, analyze those data, evaluate whether the modifications worked and the behavior of the captive parrots is closer to the wild conspecifics or not. And ultimately, we will create an EDT extension for parrots. And that's it. As a summary, we identified several improvement priorities. We proposed modifications to the zoo partners. There are exciting opportunities to encourage natural behaviors in zoo parrots. And remember, this is an ongoing research project, so please stay tuned for future updates. Thank you very much for watching.
Thank you very much, Ricardo. You do have some questions. There's a lot of hands. And I would like that you could please go through your next presentation, and with time allowing, then we can answer you can answer the questions that were posed to you from this presentation. Is that all right? Okay, thank you so much. So you have 10 minutes for the next one. So try to make some time so that people can answer so you can answer people's questions.
Now I'll be talking a little bit about the previous research project that I was involved in as part of my master's by research at University of Chester, and this is about the behavior and husbandry of mouse deer in European zoos. Introducing the mouse deer. Mouse deer are actually not mouse. They are not deer. They are an ungulate, so they belong to the family, to the order Artiodactyla, but they are in their own family, Tragulidae. They're also known as chevrotain. They're very primitive animals, and they can be found in Asia and Africa. In European zoos, we can find the lesser mouse deer and the Philippine mouse deer. The lesser mouse deer has been kept in European zoos for over 50 years, and it is a bit more common in zoos. The Philippine mouse deer, on the other hand, has only been in European zoos for a decade, and it's a bit more harder to find in zoos. The lesser mouse deer is kept under an European Endangered Species Program, or EEP. The Philippine mouse deer at the time was managed under an European Stud Book, ESP, but I think since then it has been upgraded to an EEP as well. And both programs found inconsistent breeding success across zoos. They found that both species were very understudied, and the best practice guidelines needed updating. So what we wanted out of this study was figure out what aspects of husbandry and management are affecting mouse deer behavior and breeding in European zoos. And to do that, we sent a questionnaire on husbandry and management to all the zoos. We collected behavioral data on 15 of these zoos. And what we wanted was a general study as a starting point. We wanted a larger sample size by accessing multiple collections. And we wanted a comparison. We wanted to figure out what were the successful zoos at breeding these species were they doing right in order to achieve this success? For the lesser mouse deer, we used generalized linear models because we had a bigger sample size and we wanted to know what husbandry and management was affecting breeding and behavior. In terms of main findings, one of the enclosure features that really stood out was vegetation cover. We found that vegetation cover had a positive effect on activity and natural behaviors like foraging. It had a positive effect on breeding success, and it actually decreased close proximity between males and females. And this is a good thing, and I will explain why in a minute. From a visitor perspective, we found that added visual barriers had no effect on mouse deer visibility, which means that you can add visual barriers in a mouse deer enclosure, and probably because they move around more, it doesn't affect the visitor experience. So they still get to see the mouse deer. So it's a win-win situation. In terms of water ponds, that was another feature that we found to be quite important. We found that in enclosures with water ponds, mouse deer pairs spend more time engaged in foraging behaviors. And an explanation for why water ponds and vegetation cover may be an important feature for these animals in captivity is their behavioral ecology in the wild. In the wild, these animals live in tropical forests and they move in undergrowth vegetation, which they use as cover from predators. So they are a common prey species. They have plenty of predators in the wild. So undergrowth vegetation is an important cover for them. It was also found that they are found and forage near water sources, probably because of water requirements, of young vegetation, but water sources are found to be important for them in the wild, hence why they may also be in captivity. In terms of husbandry, enrichment was another predictor that we found to be quite important and had a positive effect on foraging and moving behaviors. We couldn't include type of enrichment in the models because of sample size, but we found that the most common type of enrichment used was feeding enrichment and scatter feeding. So this one is likely to have the bigger effect. With that said, no mouse deer showed ridiculously high amount of foraging, for example, so enrichment strategies should still be revised in zoos to see what actually works with these animals. In terms of social housing, we found that most of the mouse deer in European zoos are housed socially, but that close proximity between males and females in European zoos has a negative effect on breeding. And even though we did not observe agonistic behaviors between males and females, in the wild, these animals are solitary. But male and females have overlapping territories. So it may be that it's okay to keep male and females together in an enclosure as long as they're provided with the opportunities to avoid conspecifics and to conceal themselves from conspecifics if they wish to. Diet and temperature, we found that diets were very different across zoos. We found some potential effects of browse and vegetables in the diet, and that may be because mouse deer are moose-type ruminants, and captive moose benefit from high browse, low-starch diets. From a temperature perspective, we found that mouse deer are not adapted to cold or heat, with an optimal range between 26 and 29 degrees. We could only find weak effects of temperature in the GLMs, but none of the mouse deer in European zoos were housed within the optimal temperature range. So what we want to say is there's not enough evidence from these predictors to suggest recommendations at this point, but we definitely recommend further research because both diet and temperature are likely to be suboptimal at this moment. For the Philippine mouse deer, there was a small sample size. We only ended up with 3 pairs. With that said, we found some differences in behavior between pairs. And these were more likely explained by vegetation cover, training, and food presentation in the zoos. One of the pairs that spent the most time out of sight hidden had no vegetation cover in the enclosure and spent most of the time hiding in a corner behind a little structure. So vegetation cover was likely to explain this behavior. One of the pairs spent a lot of time resting but visible to the public, so it was likely more relaxed around humans, which could be because of vegetation cover or training in that zoo. And one of the pairs spent a lot of time eating, just directly from the football. It seemed a little bit overweight, that pair. Weights were not available, but it seemed overweight, and it could be related to how the zoo was presenting the food to those animals. This is the first known behavioral description for the species, and the same ethogram was used as for the lesser mouse deer. As a conclusion, for the lesser mouse deer, vegetation cover, water ponds, and enrichment were found to be important for these species in captivity. Diet and temperature may be suboptimal, but more research is required. Social housing, they may be kept in pairs, male-female pairs, but they need to be given opportunity to avoid conspecifics if they want to. For the Philippine mouse deer, small sample size, but differences in behavior are likely to be explained by differences in vegetation cover, training, and feeding presentation. This is the first known behavioral description for these species. We provided an ethogram for both species and more evidence on a very understudied taxa. And that's it. I will leave you with a link for the paper because this study has been recently published. You can read here if you're interested. And my email contacts if you want to contact me at any point. Thank you very much for watching.
Thank you very much, Ricardo. I will now share my screen so that you can see the questions that were posed. I see a lot of hand clapping, so people really enjoyed your presentation. So thank you so much for speaking. So while that's loading, here you go. Can you see the questions?
Yes. So, did you see any effects of the human observer's presence? No. And no, because even though some of the parrots have the tendency to be attention seekers, but the way I did it, I have the pilot study where I did not interact with any of the parrots at all, meaning at least the parrots that I was studying, meaning that they habituated to me and they knew that they wouldn't get no interaction from me. So with time, they just started ignoring me, which is a bit sad, but it is what I need for a research study. So we ignore each other. And I think the visitors may have an effect, but not myself, because I ignore the animals, basically. How long was the acclimatization period after the birds were placed in enclosure before the observation and data was collected? So I haven't yet started the post-modification data collection. So these birds were already in their enclosure, and the data I collected was the baseline data. So I collected data in their current conditions. And now, because of COVID, it's been quite delayed. I was supposed to have already started a post-modification data, and I haven't. I will start very soon, I hope. And I'm giving around a month after the modifications have been applied to start the post-modification data collection. Is EDT available for wider use? At the moment, EDT can be used if you contact the University of Birmingham. The EDT is available for great apes, so chimpanzees and orangutans, and it's already being used by sanctuaries and zoo partners. To be able to use, I don't think it's a widely available tool that you just get it. However, the University of Birmingham and my supervisors who created the tool, they are always happy to discuss partnerships and to bring new partners on board. So if you are interested in getting involved with EDT, just contact my supervisors. And I can send a link in the end of the presentation to the EDT website, which provides all the necessary contacts. And I think that's it in terms of questions.
Thank you so much, Ricardo. If there are any more questions, we can see if there's some time towards the end of the conference. So now everyone will be sent into the breakout rooms that you chose.